today. COVID-19 was initially identified as a respiratory disease, but scientists now appreciate that it also affects several other organs in the body, including the heart. In fact, heart damage is a major determinant of COVID-19 related deaths, and even patients who experience only mild COVID-19 symptoms exhibit signs of cardiac dysfunction several months after recovery. A new study by researchers at the Gladstone helps explain SARS-CoV-2, the virus that causes COVID-19, inflicts damage on heart cells. And I guess I jumped right in things a little bit too quickly, but my name is Paul Lee. I'm the science education lead here at the Gladstone. And just to bring you up to speed on two housekeeping items before I hand it over to our speaker today, Gokul and Serah. The first housekeeping item is that we're going to be recording this event and posting this event on YouTube shortly after. And number two, we're going to be keeping questions or taking questions in the Q&A throughout this presentations. So we'll be keeping an eye out on that on the Q&A. So having said that, I'm going to hand it over to Gokul and Serah. Thank you everyone for showing up.
Thanks, Paul, for the introduction. Can everyone hear me okay? Yes, okay, people have raised their hand. Thanks. So yeah, thanks, Paul, for the introduction. Thank you to everyone else who's joining for this virtual lab tour. Normally, we bring you in here in person into Gladstone and walk everybody around. You can sort of get the sights and sounds of what it's like to be in one of these labs. But instead, because we're in the middle of a pandemic, we'll try to simulate that a little bit by walking you around on Zoom by holding our cameras instead. But before that, we'll give a little bit of background on what we're going to be showing you today. So Gladstone has been uniquely positioned to contribute to COVID-19 research from the very beginning of the pandemic, largely because of how many experts we have here in so many different fields, especially in the Gladstone Institute of Virology, but also in all the other institutes. So for example, very early on, the Conklin Lab here published what's called the interactome of SARS-CoV-2, which basically means they took almost every protein that makes up the virus and mapped out what proteins in a host human cell are affected by each of those different viral proteins. And since then, people all over the world actually have been referencing their study, figuring out why the virus hijacks certain proteins inside our cells and looking into potential weak points that a drug could take advantage of. Another example of a lab here is the Green Lab. One of their projects is to find ways to stop the virus from entering the cell in the first place. And the OT Lab here at Gladstone has several cool projects as well. For example, they've been able to study what exactly the virus does inside a model of human lung cells. They're also working on things like rapid diagnostic tests that might be able to tell you if you're positive for the virus within minutes instead of having to wait hours or days for your test results. So there's lots of exciting COVID-19 research going on at Gladstone, but today we're going to talk about a study that comes from a different angle, one that's definitely been understudied, and in recent months people are increasingly realizing that that is actually a huge problem, and that is looking at the effects of COVID-19 on the heart. And I'll hand it off to Serah to tell you more.
Thank you, Gokul. And can you all see me okay? So, hi, my name is Serah. As Gokul mentioned, I'm one of the scientists on this project, and we're really interested in looking at how COVID-19 affects hearts of patients. And in order to do that, we have to start with having models of the heart. Unlike, your skin or the inside of your cheek, we can't exactly take a swab of your heart in order to study it in a lab. So we need to take some other approaches. To do that, we use what are called stem cells, which you've probably heard of before. These are very special cells inside of the body that we can also create from skin or other parts of samples taken from people that can be used to generate any sort of cell in the body. So for things like the heart, which are very difficult to study normally, we can use stem cells to create models of them. And right now, I'm inside of the one of our stem cell culture labs, just to give you a quick look around the equipment and what exactly we do in here. So behind me right now, this is what's called a biosafety cabinet. I suppose this is probably the best way to see it. This is where we do all of our clean culture for stem cell research. Like the human body, stem cells can be infected by bacteria or viruses and get very sick. But unlike the body, they don't have any sorts of ways to protect themselves. So we use clean rooms or clean surfaces such as this. You can see that the blue light is on here, which is actually UV radiation, which sterilizes the inside of this hood in order to keep them protected from any sort of outside influence. I'm going to switch this around right now to show you the rest of the lab here. So these two large cabinets here are what are called incubators. This is where we leave stem cells in order to grow and then to eventually become heart cells. It's a fairly lengthy process. It takes us anywhere from a week to a month to generate them for us. So the cells that we make are very precious and take a lot of work. But we're able to do some very cool experiments with it once we manage to develop them. And so what I want to show you now is actually a few cells that have been taken out of the incubator and placed under the microscope for you to look at now. And so these are cells that originally started out as skin cells. So the same sort of cells that would be on the outside of your, your wrist or your arm. And through conversion into stem cells and then chemical treatment afterwards, we're able to turn them into heart cells or cardiomyocytes. So what you're seeing right here are in these green fibers here are the actual muscle fibers of your heart. These are what beat to create the force needed for your heart to pump blood around your body. So the kind of quivering movement that you can see here, is the actual contraction of the heart muscle, similar to what causes your blood to pump around your body. As you can tell, these are going a little bit quickly. They're beating more than once a second. So these cells are a little bit nervous since this is their first time on camera.
How many cells are on the screen, Serah?
In this screen in particular, about 1 to 200.
Wow.
Many of these cells form into tissues or areas together where they're able to contract as a unit, because each cell on its own is not particularly strong, but together they're able to generate enough force to pump all the blood in your body around. So at this point, Paul, if you'd like to show the couple of slides that we have. So once we have these cells specified into cardiomyocytes, as I've shown you, and the green here is the same thing that you were seeing before, the muscle fibers of the cells, we then, with Melanie, with the Yacht Lab here, infected them with SARS-CoV-2 virus, the actual virus that causes COVID-19. And what you can see in the second picture in the middle here, and those are the magenta pinkish dots, is that these cells are actually very easily infected by the virus. The pink in here actually shows locations where the virus is multiplying, not just individual particles, but locations where a single virus can turn to hundreds or even thousands of additional viral viruses. You can see in the bottom right, when we use some of our most powerful microscopes in order to visualize this, those small dots that you see in there are actually individual virus particles of SARS-CoV-2. And your cardiomyocytes, or the tissue of your heart, actually forms very, very many of these, more than many of the other types of cells that we've seen before. So we were a little concerned that all this virus in our hearts may actually be occurring in patients. And when that happens, that can lead to some deleterious consequences. So if you go on to the next slide, Paul. On the left here is more of the same figures that we've shown you before. And on the middle shows you what happens when SARS-CoV-2 actually infects the cells. So this is a small zoom in perspective, but you can see that those fibers, those contractile pieces that cause the heart to beat, have actually been cut up into very small little pieces, very specifically into very clear subunits that show us that the virus is in some way appears to be attacking the heart. Without those contractile, those fibers, it's actually not possible for the cell to contract anymore. So this can lead to problems where parts of your cell parts of your heart won't be able to pump blood as well anymore. So I'm going to pass it back off to Gokul now, who'll talk a little bit about ways in which we're developing methods to treat this and to prevent further heart damage.
Yeah, thanks, Serah. So now you've all seen with your own eyes this new and very concerning kind of heart damage caused by COVID-19. And the obvious follow-up question for us to ask was, what can we do about it? Can we learn why this damage is occurring? And can we find drugs that could actually protect the heart from that damage? So to answer those questions, let's first take a step back and talk about this year's Nobel Prize in Chemistry. As some of you may know, Jennifer Doudna, who's a Berkeley professor and also an investigator here at Gladstone, just won the 2020 Nobel Prize for her work on the gene editing tool called CRISPR or Cas9. How it works is you use this protein called Cas9 and direct it to target a specific spot along your DNA, like a specific gene that you're interested in. And once the Cas9 is at the gene that you want, you can use it to turn a gene off, to turn a gene on, or even to change the DNA sequence itself. That's why it's called gene editing, right? I work in the Conklin Lab here at Gladstone, where we focus on using Cas9 to correct disease-causing mutations. And specifically, my work is normally focused on engineering tools to deliver the CRISPR-Cas9 system into the kinds of cells that we're interested in. Like for mutations that cause brain diseases, we want to deliver the Cas9 into neurons in the brain, or for like heart disease, we want to deliver it into cardiomyocytes in the heart. So what does all of this have to do with COVID-19? Well, we were able to engineer a way to make this CRISPR-Cas9 gene editing system active in the cardiomyocytes, the heart muscle cells that Serah showed that we can grow in the lab. So our goal now is to use these cells that have CRISPR-Cas9 active in them to generate tons of cardiomyocytes that each have a different gene turned off. And this is like thousands of different genes, but each cardiomyocyte will have one of those turned off. And if we infect all of these cardiomyocytes together, we hope that we can find out which genetic manipulations make the heart cells less vulnerable to the virus or more vulnerable to the virus. And that way, if there's a drug that has the same effect on that gene as one of these genetic manipulations, then we might be able to use that drug to protect the heart from COVID-19 damage. Now, unlike the beating of a heart cell, which you can hopefully see with your own eyes under a microscope that Serah showed you, gene editing is a process that occurs at the DNA level, which is way too small to see. So I can't show you what a gene editing looks like in action, but I can show you what a gene editing lab looks like. So I'll also walk around like how Serah walked around in the stem cell course. So a couple of fun highlights would be behind me on this bench here is where we do a very common biological process just called cloning, which is basically when scientists want to engineer and produce very particular sequences of DNA that we need. For example, maybe it's something that we want to deliver to the cells to give them the instructions to make the Cas9 protein, for example. We need to somehow produce massive amounts of this DNA and many copies of it. So we actually use bacteria to produce it for us, like little biological factories. These bacteria, as they grow, the bacteria will produce tons of copies of the DNA, and then we just have to extract the DNA from them. Afterward, when we're done, we purify it out from all the bacterial juice and gunk using these kits behind us, and then we have purified DNA that we're able to use for our experiments. So that's just one fun site I wanted to show you. As we keep walking this way past more of these benches,
Gokul, we have a quick question before you keep going. Someone asked, what are symptoms of COVID-19 heart disease? What are some of the
symptoms?
Yeah.
Okay, so as I mentioned, this is pretty understudied so far, but there are many studies besides ours that have actually looked in patients who have either have COVID-19 or patients who have recently recovered from COVID-19 in the past like few months or few weeks. And in these people, they see functional heart defects sometimes, which means maybe the heart isn't like pumping as hard as it should be, or maybe by MRI, it looks a little bit different than it should be, maybe more characteristic of an older person than it should be. That's what we know about the heart itself. Studies like ours are looking at not the whole heart, but rather these heart muscle cells grown in a dish. And that's sort of what our study was focused on. But importantly, when we looked at heart samples from patients who had died of COVID-19, we were able to see some of these similar disruptions that Serah showed, how the fibers that allow the cardiomyocytes to beat, how they were disrupted in many places. We did actually see that, the signs of that for the first time in the hearts of patients who died of COVID-19. So we think that what we're seeing in a dish might be maybe modeling some of the early signs of heart damage due to COVID-19. That's a good question. And
Gokul, how difficult is it to access patient samples, given that COVID-19 is a highly contagious disease?
Yeah, this is one of the challenges. It's very hard to get access to patient samples, not necessarily because of how contagious it is, but more just because of how many labs want to study it. So it's very hard to get patient samples that people who have donated that tissue, but also making sure that we have access. All the labs that need access are getting access. In fact, it's even harder for us to get the healthy heart samples, which are what we would use as a control, like what we would compare the COVID-19 patient's heart to. Those are even harder to get because a lot of people die of COVID-19, but it's very hard to find a healthy heart because, you know, most
Sure.
of the people, you can't take a heart sample from a living person.
Right.
And it's very rare to find like someone who died of something completely unrelated with no heart damage and get a biopsy of that. So
Got
both
it.
the healthy and the COVID-19 heart samples are very hard to come by. And that's honestly a huge strength to being able to grow heart tissue out of stem cells in the lab. It's very transformative that we can study these in massive quantities in the lab.
Great. Thank you.
Mm-hmm. And yeah, anyone who has more questions, I see some people have been raising their hands. We can't really do anything with that, but if you type in the Q&A, then Paul and Adriana will ask your questions as we go. So as we get to the hallway, I want to point out something that I think is very unique about Gladstone and other research institutes like us, which is that all these labs are literally physically connected. So for example, Serah in the McDevitt Lab is a little bit farther down that hallway behind me. And I think this is what allows us to have these really large collaborative efforts like the ones we talked about today. For example, when the pandemic hit, we wanted to shift gears and work on COVID-19, but we're not a virology lab. But we do have expertise with, you know, stem cells and gene editing. The McDevitt Lab has a lot of expertise with growing cardiomyocytes in massive quantities, and the OT Lab had a lot of expertise in working with live viruses and dangerous viruses like SARS-CoV-2. So they were the ones who were actually able to work with the live virus in a very safe facility. So all of these studies that Gladstone is putting out are only possible because there's so much like intimate collaboration between all the different labs. And you can see that in this hallway. We're operating at lower capacity than normal due to you know, COVID space restrictions, but normally you'd see a lot of people just walking around, mingling, sharing ideas and things like that.
Yeah. One thing you showed us like down the hallways, there's no wall separating the different labs. Is there a purpose behind that?
Yeah, I think the whole design of Gladstone was intentional to that way, where like the labs are organized spatially along this hallway, but there's no walls because we're all like part of the same institute and we're all working together towards the same goal. So even like my normal thesis research is all very collaborative and all involves more than one lab, even though we sit physically in one lab. Cool. So one thing that I think is fun to look at is obviously you can't watch gene editing in action, but you can see what gene editing looks like or what our gene editing tools look like. So if you let me put on my gloves here, I've reached my lab mate Hannah's bench where she has some of these reagents. She's let me borrow them. And in this box, I mentioned that the Nobel Prize was given for discovery how we can use this tool called Tas9 to edit genes. The way that it works is basically there's two components. There's the Cas9 itself, Cas9 protein, and then there's the guide RNA. How this works is the guide RNA is something that you program. It's like a short chunk of RNA that you can custom order to match up with any DNA sequence that you want to target. And then the Cas9 is the protein that actually does the job. The guide RNA, once you get it into the cell, will bind to the Cas9 and bring it to the piece of DNA that you're trying to target and do its job, whether that's turn it off, turn it on, change the sequence, etc. So if you want to edit a gene inside a heart cell, then all you need to do is deliver this and deliver this into the cell, and they'll find each other and they'll get to work. So if you want to turn off a different gene, the Cas9 stays exactly the same. You just need to order a different guide RNA sequence. It'll show up within a few days, and then you're good to go. So that's what enables us to turn off like literally thousands of different genes and see if any of those changes can protect the heart cells from the virus. That's awesome. What we're trying to do next.
So it's almost like a word processor for the genome.
Yeah, I think that's a really good metaphor, is that if the genome was like Microsoft Word, Cas9 and guide RNA allow you to be like the cursor, where you get to click exactly where you want to make a change. And then what change you make is sort of up to you how you use the Cas9 and what kinds of tools you can fuse to Cas9. Do you fuse something that just turns off the gene right there? Do you use something that deletes a word and changes it? Things like that. So for us, we're using it in a very simple way. We just want to turn off the gene. So once we turn off a different gene in each cell, we've made like thousands of different genetic manipulations, and we can see if any of those make the cardiomyocyte somehow less likely to be infected by the virus. I see there's some more questions.
Yeah, someone asked, so you can grow cells and viruses on a petri dish to see them. Can you grow COVID-19? And if so, can you see the spikes?
This is a good question. I can answer this one. I guess I should correct it by saying it's not COVID-19 that you grow, but rather SARS-CoV-2. SARS-CoV-2 is a virus that causes COVID-19, the disease. And yes, you can grow it in a lab. However, it's a little bit harder than the sort of like bacteria and stem cell work that we do. Because it's such a contagious virus, there are a lot of safety precautions that you need to take. So for example, the lab that I just walked you through is a BSL-1 laboratory, biosafety level 1. The lab that Serah showed you was a biosafety level 2, which means it's like cells and things that could be harmful, but generally really aren't. And then there's a level above that called biosafety level 3 or BSL-3, which is where things like HIV might be studied. And then SARS-CoV-2, all of this work occurs in a special kind of BSL-3 called an aerosol BSL-3 that requires even additional training to get into. And this is why this work was only possible through collaboration with people like the OT Lab who work regularly in the aerosol BSL-3 and are able to produce massive amounts of the virus for us in a safe manner.
Just to chip in a little bit though, if you use some of the microscopes that we showed images from earlier, you actually are able to see the spikes.
Awesome. Someone else in here asked, how big of an issue is COVID-19 and heart disease? And do we know anything about the age of the people in this study?
I can go ahead and take that question if you'd like, Gokul.
Mm-hmm.
So COVID-19 heart disease is actually a fairly large problem. The estimates of how many people who get COVID will have heart disease vary quite a bit. Some people are saying that maybe about a quarter of people who become sick with COVID will eventually have some sort of heart problem, or up to almost a half. We've seen that the numbers are fairly high for people who get very sick for having lasting heart damage. The other problem is that the heart is an organ that does not grow or repair itself over time. So any sort of damage that happens to the heart doesn't heal even after the virus is gone and you get better. So any sort of heart damage that might occur from COVID has a very high possibility of lasting a very long time, even after the disease is gone, leading to increased visits to the doctor, additional medicines people will have to take in order to help support that heart. The patients in the study that we've performed span a fairly large age range. We have samples from both fairly young patients, so in their 30s and 40s, in addition to patients who are fairly old. These patients are generally above 70. So we looked at two different age groups when we went and did the study. Generally speaking, though, when we look at the hearts of very young people who have had COVID-19, the level of damage and the level of weakness in the heart that we see looks very much like someone who's much, much older. It appears that SARS-CoV-2 is having a very strong effect on damaging these hearts.
What about other peripheral organs like the kidneys or intestines or liver? Any studies about those areas of the body?
Yeah, for sure. Those are very good picks right there. Many other organs are affected by COVID. It's actually not well known how many of them become infected with the virus itself, or how many are suffering from the lungs being highly infected and causing problems throughout the body. We do know, however, that the lungs can be infected, the heart, the intestine, and liver. Kidney has had some reports of having a little bit of virus in it as well. In addition to your nervous system, there have been a number of reports and models created that show that neurons are able to be infected with SARS-CoV-2 as well. So it appears to be a very infectious virus affecting many different types of organ systems at once. That's a very good question. Thank you.
Okay. Are there any other questions for our
I see a handful in here. I can just pick through a couple and answer a couple. If Gokul,
Sure.
you'd like to do the same?
Yeah, sounds good.
Yeah, go for it, Serah.
Yeah, so I see a few questions about general techniques and what we call molecular biology or cloning in here. So I'm happy to answer those very quickly. One question asks how we remove the bacterial factories from the DNA once we produce it. And so this DNA that we produce is used in order to modify the genes of cells that we infect with SARS-CoV-2 in order to have them be more useful or more informative for the experiments that we perform. In order to do that, however, we do use bacteria as our workhorse for creating a lot of these. And so this is a method that's actually very commonly used in a lot of different drug production settings, where what will happen is you will have a bacteria that's able to produce very large numbers of whatever it is you need. In our case, it's DNA, but they're also used for producing things like protein drugs or even insulin. And once those things are produced, then you're able to use a combination of basically soap and detergent in order to wash the bacteria away and just retain the DNA. We then use these for all kinds of experiments, or these become eventual medicines for people who are sick. I also see one question about GI problems with SARS-CoV-2. This was actually one of the very first other organ systems noticed when people were mentioning that SARS-CoV-2 may be beyond just the lungs. And so many times, or initial studies have shown that when people are infected with SARS-CoV-2 in the gastrointestinal tract, so the stomach and gut, they will often have many types of very common sorts of problems, things like a loss of appetite or loose stools or other sorts of generalized symptoms. These will, follow a similar course as the rest of the disease, and it appears that so far, even though patients are very affected by this, the gastrointestinal tract is a system that grows and repairs itself very frequently. So we're hoping that once the virus is cleared, that things will return to normal. Gokul, if you'd like to take a couple.
Yeah, I can take one. I see one that asked, how did COVID-19 start? And this is something that I'd like to address because I think there's a lot of takes on it. The scientific general consensus for how it started was that it followed an evolutionary path that seems to be pretty common for infectious viruses like this, where it starts with a primary host, which is in this case thought to be the bat. Bats just have lots of viruses that naturally infect them, but these things don't normally spread to humans or to other species. They're really good at infecting bats, but not really good at infecting other things. There's usually a first jump into another animal species called an intermediate host, which in this case, we're not sure what it was, but I think some people think that it was a pangolin, which is a species that maybe these bats interacted with a lot, or maybe they interacted with bat poop a lot or something like that. This intermediate host was infected because of some mutations that the virus picked up over time in the bat that allowed it to also infect the secondary or intermediate host. And then from there, either through close proximity with these animals, or maybe there's another link with another intermediate host, but at some point, proximity to these animals allowed some mutated form of this that allowed it to infect humans as well, allowed it to cross over and infect some humans, in addition to these other species.
I see that there's another question. Does the symptom of not smelling or tasting ever go away? So as you know, one of the consequences of having this is that some people smell is affected. What do you think about that? In terms of whether it will linger or something it's temporary. Have you read any cases on the symptoms of lack of taste and their loss of smell?
Personally, I'm not sure. Actually, I haven't looked too much into the long-term effects of the smell and taste. I don't know if people have looked into that with the study and like how much they come back or not. Serah, have you seen?
So the smell and taste in particular From the reports that I've seen, generally these are some of the longest lasting symptoms of COVID, and that even after people recover, these will linger on for some time. For a lot of patients, it seems that they do eventually recover their sense of smell and associated sense of taste. However, there are cases of what we call long haulers, or people who have long COVID, who will be afflicted with symptoms or the virus itself for a very protracted period of time. And a number of labs are studying why this might happen and how to help patients who are undergoing this in order to perhaps help clear the virus or help fix these symptoms.
I have a question in here from the Facebook. What factors determine which organs become infected by COVID-19? Do we know?
This is a fantastic question, and we know some things, and we don't know some things that we're hoping to learn, maybe from things like the genetic experiments that I mentioned here. We know that the main protein that's required on the host cell to be infected by this virus is a protein called Ace2, ACE2. This virus, like the original SARS virus, has evolved to enter cells by docking to that protein on the surface of cells and using that as its mode of entry into the cells. So we know that the tissues that express, or the tissues that have cells that express high levels of ACE2 protein on their surface are more likely to be infected. But the other interesting caveat to this is that you also have to have a way for the virus to get there in the first place. So we know that for SARS-CoV-2, we know it spreads through respiratory droplets through the air. So it's very easy for it to come in contact with like your lungs, your nose, and those kinds of cells. But when it comes to cells like the heart, there's a lot of open questions on how it might get from the lungs to something like the heart, or maybe to the gut and other things. There's a lot of people have posed hypotheses for ways that the virus could travel, like maybe inside little particles, or maybe just a naked virus goes to the bloodstream or something like that. And these are open questions in the field. But generally, there are hints like ACE2 levels that we can use to guess which tissues are most likely to be infected.
A related question, is it generally common for viruses to infect several organs? Is that a normal feature we see in viruses?
Yeah, I think especially if one of the organs you're affecting is, like the lungs and the circulation, it's not, the virus doesn't have to infect every organ to affect every organ, if that makes sense. Like if you're having trouble breathing and you have low oxygen levels, naturally that's going to affect your heart and it might affect other functions as well. So a virus doesn't even have to infect multiple organs to have an effect on multiple organs.
We have someone who recently had COVID-19. She asked if there's any way to tell if her heart or other organs may have been damaged, or must she wait until problems occur to know that there are problems?
So I'm happy to take this one. First of all, I'm very sorry to hear that you've had it, and best wishes for your recovery. In terms of knowing if you have damage or what may have occurred, if you are past the disease and generally feeling back to normal, then for the most part, there isn't a real consensus from the medical community on what to do. But generally, these things are noted in patients who will have the more severe cases of COVID. So people who will go to the hospital and end up having numerous blood draws or other types of imaging tests like echocardiograms or MRIs, which will help uncover that. But we're hoping at least that patients who have had a relatively milder course, these things will eventually manifest in the form of milder or noticeable symptom, things like shortness of breath or generalized fatigue, things that linger longer and aren't normal. If you generally feel normal, though, I'm not that kind of doctor. Yeah. But if you have concerns, I would definitely suggest talking to your physician, especially if anything comes up or any new problems arise.
Right, and especially this is a new thing, and questions are being answered on an ongoing basis. And so, yeah, so this is that was a really good question from our audience members. Maybe a question for you, Gokul. A question is asked for the CRISPR research. You mentioned researching what impacts resistance. Are your labs also going to be researching recovery in the same way? And is that feasible?
That's another fantastic question, which is sort of on the hopeful side. Like we mentioned that generally heart cells don't regenerate, but what we maybe didn't mention is that the heart cells that don't die, the parts in them that are being chopped up that we showed, those do have capacity to regenerate. So our hope is that maybe if we can come up with these drugs to like save heart cells from dying, for example, maybe the damage to those cells is reversible. That's what we're hoping is the case. So we are hopeful that by screening for drugs that might be able to protect heart cells from dying, that these drugs would allow us to like stop or slow the damage enough that the heart cells that are alive can recover, is the hope. But we don't really know too much about heart recovery in general. That's just our hope.
Another question for you. Can someone with heart stents survive COVID-19?
Again, so we aren't doctors, and some of the functional studies that we've talked about were not conducted by our labs. We were just referencing those studies. They sort of motivated us to look at the heart. I can't comment on that, to be honest, but
Sure.
I can say that what these studies look at is, like Serah mentioned, echocardiograms and MRIs to evaluate the heart that way. So I can't answer that question, but I can tell you those are the tools that a doctor would be assessing your heart with to determine is there damage and things like that.
Well, on the issue of a stent or any other sort of what we call comorbidities, so these are conditions that you may have prior to having a disease that may affect your course. So this is similar to, for example, if someone who has COVID was previously a smoker and then they have some sort of diffuse lung damage as a result of that before they get the disease. So a stent is usually indicative of some prior heart condition that you may have prior to having SARS-CoV-2. And while we've seen, this is not our work, of course, but literature from the field, that having some sort of pre-existing condition definitely elevates your risk of complications from COVID, it's definitely possible for people to survive even comorbidities going on.
How much of the current research on SARS-CoV-2 has been informed by the first SARS outbreak and the subsequent MERS? 5 to 10 years from now, what do you think will be the biggest contribution of current research into coronavirus?
This is such a good question. So for those who aren't as familiar with this, the reason this is called SARS-CoV-2, severe acute respiratory syndrome, something like that, coronavirus 2, is because the first SARS was very similar. A lot of the proteins are different, but it enters through the same receptor, and it has similar, it had symptoms that were reminiscent of the first one. And they're both of the class of virus that's called coronavirus. So people naturally, when we were trying to learn about SARS-2, looked at SARS for hints. They tried first they looked at how similar are the proteins in SARS-2 to SARS. A lot of them were actually pretty different. So unfortunately, we couldn't take that much over. But a lot of them do have similar roles, and by comparing and contrasting, people could learn a lot. And to the second part of that question, now that we have the tools, a lot of labs are doing cool studies where they compare not only SARS-2, but they also do similar studies with some of these other coronaviruses like MERS, SARS, and even other viruses like influenza. And they compare what is the same across different viruses, what is the same across different coronaviruses. And we're starting to learn more as a field about which of these effects apply to coronaviruses or to viruses in general, or to only to SARS-2. And hopefully 5 to 10 years from now, if there's another similar outbreak, maybe we'll be able to draw on things from SARS-2 or from other viruses as well.
Yeah, hope so. So what is it like to work in a lab during the COVID-19 pandemic? Are there extra guidelines you have to follow, such as fewer people in the lab? And does that impact the speed of the work you can do? So maybe you can show us some of the PPE that you are using over there.
Sure. So I'm currently in a private office right now, so I have my mask off, but there all of the things that you said are true. There are extra guidelines in terms of the number of people that can be around each other at a time, in terms of the number of people in the lab, in addition to when there are people who aren't able to maintain a certain amount of social distancing, we have a number of different precautions in place. These include face shields to prevent from transmission of droplets. So from people talking or coughing, these plastic screens protect against that coming in or out. In addition to all types of different masks that we wear all the time. Gokul is showing one off right now, which is the general use one that we use most of the time, although there are more specialized, more protective masks used for people who are working very closely. For the virology labs who actually handle the live virus in the aerosol BSL-3, as Gokul mentioned before, there's an even higher level of protection necessary. And I also wanted to mention that, in addition to the protective measures that the individual people are taking in policies, there's been a lot of effort on the part of the facility and building in order to maintain safety as well. For example, there's a much higher level of airflow now in order to make sure that air is very rapidly vented out of the building to prevent things like the virus floating around and circulating and infecting people. So there are lots of precautions in place. It is a very different environment now, but it has kept all of us here very safe.
That's pretty comprehensive. And I think another thing I'd like to emphasize is a lot of us on our own volition, but also because Gladstone makes us, we get COVID tests very regularly. Being in San Francisco, this is something that's very available to us. But I know people in other parts of California and even Arizona have these, where my family lives, have these tests pretty available. And I think regular testing is something that can not only put your mind at ease, but also help make sure that if there was someone who tested positive, we would know, and we would know who was in contact with them, and we'd be able to minimize the spread if that ever happened. So that's another important safety thing that anyone can do, no matter where they work, is just take advantage of any free testing or COVID testing you have around you.
Yeah. Well, thank you for doing what you're doing and keep up the good work. Another question we have from our audience is, I know there's been a lot of talks on a vaccine, but are there any research that's being done on treatments, such as, for example, HIV has never had a vaccine, but after a long time, treatments were developed. So maybe one of you can speak to that?
Yeah. In fact, one of the hopes of our screen is that we might be able to identify a cardiac-specific drug type of treatment that could maybe alleviate some of the heart effects. But that's, again, this is only a small slice of what's going on. Even at Gladstone, there are people looking in the lung, in lung cells, and trying to screen for drugs that can either reduce infection or keep cells alive when they get infected, things like that. From a lot of different angles as well. There's just brute force, try thousands of different drugs and see which ones protect them. There's a more, like I mentioned, the interactome approach from the Conklin Lab. They are looking for, if they can find certain host human cell proteins that the virus needs to hijack, then if they can disrupt that interaction, then maybe the virus can't survive anymore. That's another approach that people are taking. And often they're taking this approach with what's called repurposed drugs, which they're not trying to make a new drug from scratch. They're trying to see if there are drugs that have already passed, phase 1 clinical trials for safety. And then maybe they just were abandoned because maybe it was a schizophrenia drug, for example, that didn't end up curing schizophrenia, but it was still pretty safe. Then maybe you could, if it happened to have a side effect now of reducing the effect of COVID-19 in the lungs, then maybe that's the drug that could be fast-tracked to wide distribution. So these are some of the approaches that people are looking at besides vaccines.
Mm-hmm. What about immunity? What determines how long immunity lasts, either from having the virus or getting a vaccine?
That's a very good impression question. There are a lot of factors that go into it, and many of us here we're not vaccine design specialists, but there are many different things that go into it, ranging from the strength of your own immune system prior to getting that vaccination. For example, older patients generally will have a repressed or depressed immune function, and so will need a stronger or additional amount of vaccine material in order to mount enough of an immune response to have enough antibodies to fight off an infection. In addition to that, many things about how the vaccine itself is created, what parts of the virus it uses in order to immunize the body against infection, in addition to other aspects such as how quickly the virus mutates. This is a question that's come up quite frequently, and I think it's worth mentioning now that the SARS-CoV-2 virus, there's been a lot of worry about it mutating and evading a vaccine. However, from our knowledge, at least, that it appears to mutate rather slowly, and so we have, similar to what Dr. Fauci had said, we have no reason to believe that an effective and strong vaccine against COVID won't be effective for at least a couple of years. And of course, there are other ways in order to modulate that immunity to, generate or to help people who are otherwise at risk or immunocompromised. Some of the labs here, including ours, are looking at ways to perhaps, boost the immune system of aged people who are the most at-risk population for COVID.
I know for the study that you were a part of allowed collaboration between labs within Gladstone, right? So a question asked, how much have you partnered with other institutions, either to do research or scale up production and distribution of possible therapeutics? Anyone would like to speak to that?
A lot. I think every study that even the study that I mentioned, but also the studies that I shouted out from other people like Gladstone, I think without fail, every single one of these has been worked on by people at other institutes, whether that's UCSF or Berkeley or even people in other countries. I think COVID-19 has forced people to work together more. Part of it is because, not as many people from one lab can even be in at the same time. So you almost have to collaborate with other people to make it happen. But also the reason a lot of these studies have been accomplished so fast is because people are just willing to work on them wherever they are in the world and work together more than before.
I think there's a lot to be said for that collaborative nature. I saw a handful of questions about Gladstone itself, and I just wanted to mention real quick, the virology groups here at Gladstone, including the groups that Gokul mentioned at the beginning of the talk, Nevin Krogan and Melanie Ott, they were very rapid adopters of model systems for COVID-19. Nevin's lab in particular actually started research on the disease back in December when it was just isolated to a few cases in China. And so Gladstone's been able to pivot mainly because of, the collaborative nature of, labs such as that who have done a lot of work in generating the foundation for this sort of research, and then being very generous with their time and resources and sharing it with other members of the community.
Yeah, audience member, just more of a comment than a question is amazed by how quickly Gladstone and other institutions were able to pivot from one problem to another, and just how fast other and many other organizations have been able to contribute to helping during the COVID-19 crisis. Adriana, do you have any questions from the Facebook side?
Yes. Is the heart affected only after the patient recovers from COVID, or is this something you can predict while the patient is getting treated for COVID in a hospital?
So it's a great question. Thank you for asking. I think the best way to answer this would be to step back into a little bit of history. Back in about March and February of this year, this was actually how we discovered that patients were having heart complications from COVID. Very much people knew that the lungs were being affected from previous research in SARS-1, but when people were taking blood draws in the clinic when they were being treated for COVID, during those blood tests, they were doing assays for all sorts of markers in the blood to, tell the doctors, what sort of things are going on with your body. And in addition to the normal things that you would look for in an infection, some of those studies began looking at markers of cardiac damage. There are a couple of very, very good markers that show when the heart is being damaged in some sort of way that people were seeing were going way up in the cases of people with COVID, but way back then. So in a way, we actually discovered heart damage in COVID through prediction in clinic rather than the other way around. It was only after a few months and a lot more recovered cases that we began to notice people with, longer lasting or manifesting after recovery symptoms.
We have an earlier question that asks if there are any any opportunities to get involved with Gladstone as a citizen scientist with regards to COVID-19?
That's a good question. Obviously, a lot of this research has been funded by donations, but I would say in terms of citizen science specifically as a way to contribute, I'm not sure how much Gladstone in particular has done, but I know there are generally a lot of COVID-19 citizen science things out there, whether that's about
Sure.
what's it called? Tracking, contact tracing and things like that. And there's some cool, the structural protein people have some cool citizen science things where they can crowdsource possible solutions for a new drug that might fulfill a certain shape that people do through just online, almost like games. And a lot of COVID-19 researchers have been taking advantage of that sort of citizen science approach. I'm not familiar with what Gladstone in particular has been doing in that space though.
Yeah. Well, we do have a philanthropy team, and so I'm sure we'll be happy to accept any donations to our science and to our research, especially during these times. So it's another way to contribute to our science.
I was going to address one of these questions here, which I think is great. Just pulling back a little bit. So what do you really do in your lab? When you go into the lab day to day, what are you actually doing?
Do you mean before this or after this or during this? I think Gokul alluded a little bit to his prior research, but I think we can both give a quick. Okay,
Yeah.
someone just asked.
I think it's more of the day in the life of a scientist.
Okay.
Yeah.
Geez. It depends a lot on what we're doing. A lot of it is upkeep. When I was in the tissue culture room and showing you around, we have many cultures going on at a time. And so a lot of what we do on a day-to-day basis is maintaining and taking care of those cells, monitoring them for quality and for behavior so that we know that when we use them for experiments, whatever we need them for we can be confident in whatever results we end up getting. In addition to that, big days in the lab or include times when we're organizing large experiments that actually involve live virus. It is a collaborative, coordinated process with multiple labs in order to do that. And so usually it'll be about 2 or 3 of us who are in here at one time spending hours a day, sometimes starting from very early in the morning. I'm not a morning person. In order to set these experiments up. So this includes a lot of time in those biosafety cabinets that I showed you, a lot of time going in and out of the incubators and on the microscopes. In addition to, after being in lab, a lot of the time we spend at home is often spent in meetings or looking at data that we've acquired and analyzing it.
Gokul, would you like to add to that?
Yeah, I would add that I think something that people don't realize is that a significant fraction of our time every day as a scientist is spent in meetings. Nowadays, they're all virtually over Zoom, but even previously, many times a week, we would have meetings of some sort, whether that's a one-on-one meeting with your advisor to talk about your research and give updates, or a lab meeting with members of your lab coming together to give you feedback on your work. Or at Gladstone, we often had these things called RIP seminars, or research in progress seminars, where you present to other people on your floor and other people in your institute at Gladstone and get feedback from the broader community that way. So a lot of our time is spent, giving each other feedback, building those collaborations that way, and doing a lot of interpersonal interaction and thinking. Besides that, a lot of time is spent, at least in the area of work that Serah and I both work in, which is stem cell based work. A lot of time is spent keeping our cells alive and keeping them happy. Or if we want to make them into cardiomyocytes, that's a month-long process to guide stem cells that can turn into any cell in the body, guide them directly down the path to become cardiomyocytes, or in my case, maybe neurons for some of my other projects. Mm-hmm. So a lot of these things take up a lot of our time. We have millions of little children we have to feed the lab.
I'm sure.
That takes up a lot of time. And then some of the, we mentioned the molecular biology work, meaning engineering pieces of DNA that you want to deliver to the cells and stuff like that. That stuff is a little bit more flexible, but I'd say we spend a lot of time in meetings and we spend a lot of time feeding ourselves.
Great. Well, as our last question for today, more like a fun question, what got you into science? Were there any specific mentors or teachers from high school or from college, or were there a particular class that got you into becoming a researcher and a scientist? And maybe share a little bit more about the personal side of how you got to where you are.
I have a funny story about this, actually. So for a while, I wasn't really sure what I was going to do. And it was when I was in high school, actually, when Shinya Namanaka won the Nobel Prize for the invention of induced pluripotent stem cells, or, well, shared with, I believe Dave Gordon. But when that Nobel Prize came out, and I heard about what was going on and the potential of these cells, I thought to myself, like, this was such a fantastic and wonderful opportunity to do a lot of really amazing science. And so the drive to begin to study those and to use those and to help people and to advance medicine was really the thing that was pushing me forward. The funny story part of that is that Shinya is actually an investigator at Gladstone. His office is about 4 doors down that way. And when I first joined the lab here and began working, my very first interaction with him was kicking him out of a conference room because he was over his reservation. So never meet your heroes.
Wow. Gokul, would you like to have the last word?
Mine was a little bit less dramatic than that. I think in undergrad, I was fully intent on becoming a chemical engineer because, I don't know, I had made decisions on like what sorts of classes I liked the best. And I think chemical engineering was a major that I decided on. But then freshman year, I took a seminar called Careers in Biotechnology because my school made me take a seminar in something other than chemical engineering to see what else is out there. And I learned about, some professor gave a talk on some stem cell based research where they were applying stem cells to spinal cord recovery, where neurons that don't grow back normally, they were able to create biomaterials and certain stem cell based transplants that were able to like restore motion in people who used to be paralyzed. And I think that introduced me to the power of these biological things like stem cells. And eventually I joined a lab as an undergrad and started researching, and I was exposed to both stem cell based research and gene editing based research. And that's something that stuck with me. And I knew throughout college that's what I wanted to pursue later in grad school.
Great. Well, thank you, Gokul and Serah. We're at the top of the hour, and I would also like to thank all of our attendees for attending our virtual lab tour event. Please enjoy the rest of the event sponsored by the Bay Area Science Festival, and hopefully we'll see you guys around. Take care. Bye-bye now.
Thank you, everyone.